thanks a lot for giving us a chance to present this paper here, actually. This is a very relevant audience, so I'd be happy for any feedback. Please just interrupt whenever you want during the talk or afterwards. Also, I'll start with a presentation, and then in the middle we will switch. So Joao will do the second half. So we'll have a quick break, maybe also for discussions. All right, so this paper, as Philip has already said, is a review paper, which has a topic of schizophrenia, and we want to understand working memory in schizophrenia. And the method of choice that we use to understand working memory behavior is this class of attractor models that can model the tasks that we're using in patients. So this has been a work with Compte, who today is not talking, but he's been very important in this. So hi, Albert, if you're there. Let me see. Okay. In schizophrenia, we know that that certain working memory deficits persist in this group of patients. So usually those have to do with precision, but also schizophrenia patients are more distractible than healthy controls. They show reduced capacity, and as we have recently shown, also serial bias. So here, for all of these measures, we see that often these deficits are delay dependent. So they actually stem from working memory. This is the case for precision and serial bias, and possibly also for capacity and distractibility. When we want to understand these behavioral deficits in the context of attractor models, then we first have to start thinking about how these attractor models solve behaviorally these working memory tasks. And an important starting place here is to look at neurophysiological data of monkeys who are performing such a spatial working memory task. So here in the late 80s, Funahashi and colleagues did this experiment in the Goldman-Larkich lab. The monkey had to remember one position on the screen. So here you can see the different positions that have been shown. And at the same time, they recorded from single neurons of the prefrontal cortex. And importantly, what they found in these recordings was that specific neurons had preferences for specific locations on the screen. So here, this neuron whose rastergrams we're seeing here prefers the upper left location. And more than that, these neurons that prefer these specific stimuli also spike throughout the whole delay period. Okay. So the two important features that we have in this task is at one hand, stimulus specificity, and on the other hand, persistent delay activity. So this is the starting point for what we want to achieve with a computational model. And computational scientists who have started thinking about this in the early 2000s usually use this kind of ring models that we also know from, for example, head direction cells. So here, the basic idea is that neurons in this model are ordered by their preferred location on the screen. And then as a second ingredient, we have a specific connectivity profile where neurons with similar preferred locations are more strongly connected than neurons with dissimilar locations. And so what these models achieve is that activity is passed back and forth between neurons with preferred similar locations. And through time, these neurons will keep firing and they hold the memory throughout the whole delay period. As you can see here in this bump of activity that persists through time. So importantly, when we want to understand the behavior of this network, we just go to the last time bin of the simulation, and then we can read out the population vector and decode the remembered location. So another thing that monkeys show in this working memory experiments is imprecision in working memory that increases with the length of delays. So here we have higher standard deviations around the targets of the responses for very long delays. And again, this is a finding that these attractor models can explain very elegantly. Specifically, we have to look at the drift or actually at the diffusion of the center of mass of this bump over the course of the delay. So when we measure this in several trials, and what we see is that the bump diffuses to some random location in each trial, to a different one, and the diffusion increases, or the spread of this cloud of locations increases as the delay gets longer. So when we want to understand the behavior of the model at 0, 3, and 6 seconds, we just go to the specific time points and read out locations here. So I'm not going into detail about this, but these models have also been used to explain distractibility through other stimuli, but also limits in capacity. And as we have recently shown, also they can explain serial dependence between adjacent trials. So going back to the deficits that we see in schizophrenia, we can again cluster them by these 4 different measures. And we see here in blue the specific, well, it's only schematics, but these are the specific response patterns that we expect to see for schizophrenia. So we have decreased precision, higher distractibility, lower serial dependence, and lower capacity. And all of these findings have been modeled in such bump attractor models of working memory with different biological alterations in the network. So here I'm just showing you a few of them. One of them is EI ratio that is usually modulated by decreasing NMDA receptor functioning at inhibitory or excitatory synapses, resulting in increases in excitation or decreases in excitation in network. Then similarly, there's been some accounts of dopaminergic modeling of these altered attractor dynamics. So specifically, it's important whether dopamine 1 or dopamine 2 receptors predominate the picture. And finally, we have recently shown, and other people have too, that short-term plasticity affects the way that, for example, bumps diffuse throughout the trial. So this multitude of biological mechanisms and how it affects attractor dynamics can be understood through by looking at the attractor landscape that determines how this task, how memories behave during this task. And one, yes?
Sorry, just a brief question. So for the dopamine part, I mean, that's essentially a neuromodulatory mechanism. So it might not be, you know, mutually exclusive with the change in excitation inhibition ratio, right? So you could imagine that, you know, the dopaminergic tone, or depending on which receptor you predominantly target, that it changes basically through the second messenger cascades, you know,
Yes.
excitability of the neurons, or something like, you know, NMDA receptors or so. So how did those studies that looked at these D1, D2 ratios actually model this at the biophysical level?
So basically, they model this as an effect on a lot of variables, right? So they don't only switch one part of the synapse, but rather this has a, as you just said, it has a cascade of effects, and maybe we're presenting this here as 2 very different mechanisms, but in fact, some people have argued that EI ratio could also be just modulated by D1, D2. So yeah. Here, when speaking about EI, I mostly mean NMDA receptor-based changes, but yeah, it's not exclusive in any way. And also, short-term plasticity might depend on these other factors as well, so they're not independent mechanisms for all of them. Oops, okay. So yes, this might be a familiar picture to several of you. So this is one energy landscape for specific attractor models. So here we have a double well energy landscape that often is used to think about schizophrenia. So this is from the Dursevitz paper. Actually, they modulated D1, D2 ratios, but we could also think that changes in EI ratio might determine the shape of these energy landscapes, right? What these schematics are showing is that as we change those biological variables, we obtain more stable or less stable attractors. So depending on the depth of these wells, our memories will tend to more easily switch to the other attractor state, or they might have more difficulties. So the more profound this well, the more difficult it is for the memory to be switched, right? And you have seen, I think in the last Runner Club, I haven't been here, but I know that you discussed this paper from the Murray group, where they have applied this kind of double well model to discrete decision making. So basically they have shown several perceptual samples and asked subjects to make a decision. And what we can see here is that for elevated excitation in a network, we get more recency effects. So this is just reflecting the fact that we fall into one attractor, and then the attractor being more stable than the other, we stay in this attractor longer. And the contrary is true for lower DI ratio. So now we wanted to ask whether this is also a relevant framework for thinking about working memory, and specifically continuous working memory. So as a spoiler, I'm already giving away that this is not, of course, not the right model to understand continuous tasks. And I want to exemplify this in the paper from John Murray from 2014. So what John Murray tried here was to, or what he succeeded in, was to model increases in diffusion of memories and also increases in distractibility in schizophrenia by increasing excitation in a network. Well, actually what he did was to decrease inhibition, but this results in increased EI ratios. And the effect that this has on the neurons of the network is that more neurons start to participate in such an activity bump. And on average, the firing rates within the bump are also higher. So one might argue that, in fact, in this disinhibition case, our attractors are more stable, right? Because their wells, or the attractors are deeper, we have stronger firing rates, and we would expect memories to possibly diffuse less. However, what they found in this paper was that diffusion was in fact increased for the disinhibited condition of the network. So what we wondered here was why would increased attractor strength lead to more diffusion? To understand this question, we have a class of models, which is more suited to this task. In retrospective, this might look obvious, but to model tasks with continuous stimulus spaces, we have to use continuous attractor models as a framework. And here, the effect of increases or decreases in EI ratio is actually most strong on the walls that are orthogonal to the manifold on which memories lie on. So to explain this in a little bit more detail, the different spatial locations that are being held by the network lie on this pink manifold here that you can see. All of those different states have the same energy for each one of those wells, right? In contrast, where the walls actually get steeper and where the walls are more difficult to be overcome by noise, those are directions that are not relevant to this memory manifold here. And so when we notice that that deepening attractors doesn't actually prevent noise from affecting the drift here so much, we wanted to, we started to wonder, what does diffusion in this kind of model actually depend on? And switching over to Joao.
So there is these papers that Albert found where they studied these bump attractor models with this mathematical framework called the mean field analysis. And I'm not going to derive this, and I wouldn't know actually, but there is this very compact formula that really explains what's going on here. So what they show is that the diffusion, or this, how much the bump drifts one side or the other. So this diffusion parameter depends on these three factors. So it depends inversely on the attractor strength. So actually having a stronger attractor should lead to less diffusion, in contrast to what one would interpret by the figure we just saw by Murray et al. But it also depends on the noise. It depends on the noise in two ways. It depends on the input noise, so that each neuron receives inputs from many neurons, and you can look at how this how variable this is relative to the mean throughout a given time, let's say 1 second. So that would be the input noise. And also how this noise is correlated. So the correlations between this input noise, how much this noise is correlated across neurons. Okay, so these two factors affect diffusion in, like, makes the bump more diffusive. So it would be less, the performance would be worse in a task, and the attractors make it better. So to show this, to show that it's more complicated than just look at the attractor strength, we have these two models. One is this classical model that has been used by Murray and others, and we have this alternative model, which is less popular, I would say, in computational psychiatry. And we don't, I just want to say straight ahead that we are not saying that one or the other is more plausible. We actually, to make our point, we want to say that we could reasonably choose one or the other based on the following characteristics. So the first model has symmetric connectivity and all-to-all. So it's not very biological that we think that all neurons in this network are connected. And they have this very, very specific shape, this cosine tuning. So all neurons in this network have the same tuning. They are just a bit shifted. They are tuned to different stimuli, but they are exactly the same neuron. While on this other model, the connectivity is pretty random and sparse. So you can see that a lot of white space is here. So it's not all-to-all. Actually, each neuron receives just input from a small fraction of neurons, while here it receives the input from all neurons. And so this leads to the noise in this case is internally generated by this randomness in the connectivity, while in this case the noise is generated by an external input. So we stimulate all the neurons in this network by a very strong external input that is noisy, and that's how noise arises in these simulations and in Murray simulations. And so this leads to homogeneous tuning curves, as I said. Well, in this case, the tuning curves are heterogeneous. I don't have a figure for you, unfortunately. And there are other details that might be important or not for what we are going to show. So the synapses are conductance-based, while in this case they are current-based. The NMDA receptors are more biological, so they saturate, as in real neurons, while in this case they don't saturate. And both of them have short-term plasticity. So all I want to show with this slide is that you could think, okay, this model is more biological because it has a random and sparse connectivity. But on the other hand, this one has conductance-based synapses and saturating NMDA receptors. So I think it's fine to say that these are on more or less equal grounds in regard to biological plausibility. But they lead to really opposite predictions when one tries to do this manipulations that Murray does. So what we did here, it's in both models, we increased and decreased the I ratio by increasing excitation to excitation or increasing excitation to inhibition. But basically, we are increasing or decreasing the I ratio in both models. And what we see is for the Stein et al. Or for the Murray model, we see what they saw. So it increased diffusion as you increase the I ratio, and it decreases diffusion when you decrease the I ratio. So it's the same finding as we saw for the review that Heike made. But on this alternative model, we see exactly the opposite for the same kind of manipulation. So this raises the question of what's going on here, and somehow it calls for, right now it calls for a caution to interpret this as like a model of schizophrenia, because you could have chosen another model that would be equally plausible, and it gets the complete opposite prediction. So what I'm going to do now is unpack this formula. So each of these factors, I'm going to analyze them in the model. And so we see that for the Stein et al. Model, there is a slight increase on the bump as you increase the EI ratio. As you decrease, there's a slight decrease. While for the Hansel and Matteau, you have a strong increase of the bump with an increased EI ratio. So more or less the same direction, but this one with more dramatic changes. On the other hand, when you look at these two types of noise, you see that the input noise in this model changes as you increase or decrease the I ratio on the same direction as the activity. While for the Ansel-Matot model, it doesn't change at all. On the other hand, if you look at these noise correlations, you see that one very dramatic difference is that because of this sparse connectivity, the correlations are much smaller, much weaker than the one with the all-to-all connections, which makes sense. Yeah, all the neurons are connected, so you have much stronger correlations. And then if you look at this amplitude, which is what goes into the formula, you see that there is an increase of this correlations as you increase EI ratio, and a decrease when you decrease EI ratio. This could be bad news, or you can turn around and try to derive experiments to test in which regime the cortex is operating. And so I'm just going to finish this presentation with some suggestions or some desires that would be nice to see this data somewhere. So for neurophysiological experiments, it would be nice to see with and without pharmacology. Of course, ketamine is one of the best candidates for pharmacology to test this hypothesis. And so we think it's important to characterize tuning curves during working memory, as opposed to what has been done in the field, just looking at average fine rates or baseline activity. Also, noise correlations, however, as Toby was hinting on, these plots are not for spikes. So of course, we need to repeat these plots looking at spike counts instead of currents. And of course, how these changes correlate with the impairment that you see in behavior when you use this pharmacology. On the other hand, I think just purely behavioral experiments will be really valuable to constrain more the current models of working memory, schizophrenia. We have done this comparison between schizophrenia and NMDA encephalitis, and we think it's a powerful way to compare two populations. But also, of course, with pharmacology, once again, ketamine, and yeah, this was just last minute. I think there should be some care to generalize the intuitions we have from discrete tasks and this discrete attractor when we think about working memory tasks or continuous attractors. There is some care that should be taken. And then there is this, I want to mention this paper that Heike already mentioned, where they were able to find this, like in a very large data set for schizophrenic patients, but it would be nice to see the same with ketamine. So they had a task where they could study diffusion and distractibility. And we think it's possible to have a task with just some minor changes, a task that you can test all of the impairments that is known the schizophrenic people to have, and even other biases. And they found in this paper that the distractibility and diffusion correlate very strongly. So it would be nice to see that also the same for other working memory impairments. This would mean that they are measuring something relevant for schizophrenia. And yeah, I think that, oh, there's no more slide. I think that's it. I had a thank you slide, but for some reason it doesn't appear. But yeah, I want to, there you go, very important slide. And I want to thanks Albert. I mean, all the credit goes to him, and all the mistakes that we presented today, it's on us, of course.